Great, and now I'm going to introduce for our next session, Dan Hartman, Director of Integrated Development for the Bill and Melinda Gates Foundation, who's going to chair this session on the use of big data and capacity development as drivers for future innovation. And I think a number of colleagues will join him on stage. Thanks, Dan.
I'll ask our panelists to join me up here. So I'd like to thank the organizers for inviting me to chair this session. I'm Dan Hartman. I'm the Director of Integrated Development at the Bill and Melinda Gates Foundation, which is nondescript, but what it really means is my team provides technical expertise in the area of regulatory manufacturing and pharmacology to our malaria team, and also supports our partners like MMV. In addition to being the Director of Integrated Development, I also had the pleasure of serving as interim director of the malaria team, so I got to really learn the community and work closely with MMV, and that was really a pleasure. So I'm privileged to represent the Gates Foundation here at this celebration. The foundation has been an enthusiastic supporter of MMV for many years, and for the last 10 years under the leadership of Janice Culpepper, who's somewhere out there, and many of you know her, and she's just done a fantastic job managing this relationship. Personally, it's been a pleasure for me to work with David and the leadership team going on 8 years, and I've seen a lot of positive change in the organization. They've built the necessary capabilities and capacities over this time to really substantially contribute to the malaria eradication effort. So this session's about knowledge sharing, big data, capacity development as drivers of future innovation, and I'm joined by an exceptional panel, and I'll share a bit about each one of them before I ask them to talk, just so you can get a sense of their backgrounds and how credible they are. I'm going to start with Jeremy Burrows to my left. Jeremy's the VP and Head of Drug Discovery at MMV. He leads their discovery team with a goal of developing new candidate drugs for anti-malarial therapies, anti-relapse, prophylaxis, and transmission blocking. He obtained his master's and PhD from Oxford. He subsequently joined AstraZeneca, where he worked on a whole host of different disease areas, cardiovascular, inflammation, and even Alzheimer's. In 2008, he joined MMV, and in 2010, he became the head of the discovery team, overseeing a growing portfolio, which has been talked about this morning, of enabling technology, screening, hit to lead, optimization, with a variety of different partners, many of you sitting in the audience right now. He's a fellow of the Royal Society of Chemistry, an honorary research fellow at the Liverpool School of Tropical Medicine. He also has on here, he enjoys walking in the Scottish Highlands and drinking whiskey, so I think those two might go together. So anyway, Jeremy, please.
Dan, thank you very much. So there is an urgent need still for new antimalarials, either to combat the future resistance associated with current treatments, or indeed to address the problems of prophylaxis, transmission blocking, and anti-relapse in ways beyond that which we can achieve now. So it takes between 3 and 5 years to deliver a candidate drug, and at least a further 10 years before a combination product is available to patients in Africa. So the need to pioneer new efficient, rapid, cost-effective ways to do drug discovery is critical. So how have we tackled this over the last decade, and what does the future hold? So first thing to say is that the MMV discovery team is small. 15 people based in offices near Geneva Airport cannot achieve very much on their own. And indeed, our first innovation has been to collaborate, to build a portfolio of global projects, selecting only the best partners and projects. And indeed, collaboration is our lifeblood, and it is such a joy. In fact, it's really quite overwhelming to see so many partners, past and present here today. And I just want to start by thanking all of you for your contribution to MMV's mission. Thank you. So second, the combination of technological advancements, such as the improvements in high-throughput screening and driving down the cost of screening, in combination with the outstanding leadership of our partners, has led to over 7 million compounds being screened against Plasmodium falciparum. By my reckoning, this is the single greatest concerted screening campaign against an organism in history. And as you can see on the photographs, Elizabeth Winzeler, Vicky Avery, Kip Guy, and Javier Garmo and their teams have been giants in this area. Screening has also been productive, delivering over 16 candidate drugs. So third, we've built open innovation modus operandi. With the help of our business development and legal teams, every new partner that joins into the MMV Discovery mothership gains access, complete access to the full constellation of platforms set up as centers of excellence by our partners. Now, these assays are validated for quality, for throughput, for cost effectiveness. And since key compounds from our projects are tested through multiple assays with the invaluable help of our project coordinators and data manager, this gives MMV and our ESAC the ability to compare projects with standardized data. And this provides an inherent quality standard through all our projects and the associated literature. Open innovation also actually provides a framework in which there are many people in this room who are collaborating who otherwise would never be talking. Now, I think that's something to really celebrate. Fourth, knowledge squared is knowledge shared is knowledge squared even. So over a period of 8 years, over 20,000 compounds and associated data have been uploaded into public databases. With committed partners, a machine learning model even has been built using the screening data from over 5 million compounds and associated data. And this model allows us to predict completely new compounds active against blood-stage malaria. This tool will shortly be available on the Chibale website and is the first in silico tool for malaria of its kind. Now, MMV is committed to sharing learnings, and through our publications and through our input, project teams are able to gain the clarity and criteria to actually help maximize effect in the field. So lastly, there is still much to deliver, and we must never be complacent and constantly challenge the way in which we work. So big data will undoubtedly become more and more important as the amount of structural, biological, target, phenotypic, genomic data becomes available, increases in volume, extent of annotation, and public disclosure. This actually gives the opportunity for increased new machine learning techniques, as well as exciting new target, phenotypic, and open source drug discovery approaches. And this future, in collaboration with our partners and an ever-evolving open collaborative environment, fills me with great hope that we collectively can fulfill our part of MMV's mission to discover new antimalarials for the world's poor. Thank you very much.
Great. Thank you, Jeremy, for that, that presentation. And it really, I think, did highlight the new era that we're in of big data, about how we can look at data, how we can share data, and how we can make advances. And your work has clearly demonstrated that. So looking forward, what do you see as some of the biggest challenges in the discovery area, and how can we overcome these challenges?
So I'd really break the challenges into 4 parts. As I mentioned, we've done a lot of screening, 7 million compounds against blood-stage malaria. So one of the key challenges for us is, where do we find new compounds? A second challenge is in relation to the other life cycle stages, particularly hypnozoites and Plasmodium vivax. How to find new, attractive chemical start points for discovery projects. A third challenge is how do we ensure that what we actually deliver is going to produce the maximum impact needed in the field. And the fourth really is our drive, the challenge to be as effective as possible in what we do.
Great. Well, thank you very much for that. Our next speaker is Professor Elizabeth Winzeler from the University of California in San Diego School of Medicine. There, she leads a group that uses systematic, data-intensive methods to solve problems at the interface of host pathogen biology, typically involving large collections of chemical screening data and whole genome sequencing, as we've seen and she's talked about already on this stage today. She's the winner of numerous awards from including the Keck Foundation, the Ellison Medical Foundation, and even the Bill and Melinda Gates Foundation. In 2014, she was awarded the Bailey Ashford Medal for Distinguished Achievements in Tropical Medicine, and in 2017, the MMV Project of the Year Award. So, Elizabeth.
Thank you. So I'm someone who works a little bit at the basic research end of things, and I really want to emphasize how important it is to continue with basic research, because I think we've learned just a tremendous amount. So I'm going to give a couple of examples of how we both generate big data, and Jeremy alluded to how we use a lot of high throughput screening, and then I'll give some examples of how we can use these larger data sets to accelerate the process of drug development and to learn new things about malaria. So, actually, I'm not sure where the, okay. So I'm going to be talking, so I'll just show my first slide. So my laboratory, and as well as partners, we're very involved in developing new phenotypic screening methods. And so we've developed screening methods that predict whether a compound has chemoprophylactic activity, whether it can relieve symptoms of the disease, and potentially also whether it can block transmission. And working with MMV, we've gone through literally probably about over 1 million compounds over the last 6 or 7 years. And all of this data that we're creating, we put into public databases, and not everyone has the tools that we have at the University of California. So tools for doing liquid handling, for dealing with really, really small amounts of compound. And what we want to do is put all of the data that we create through these screening methods out into the web, so that if someone is interested in advancing a molecule, they can take the data that we create in our laboratory and use this as a starting point for chemistry and lead optimization. And I just wanted to show this one slide. It's got a lot of details in it, but this kind of highlights the advantage of doing everything in a very consistent way. So we recently were involved in a screen where we obtained the Charles River Library from MMV. This was in collaboration with Brice Campo. And we screened 1 million compounds, testing each one of these for whether or not they could inhibit the liver stages of the parasite. So we were looking for new compounds that could potentially be used in chemoprophylaxis. And we can take all of this data, and what was very cool about this is that when you have access to everything, you can take all of the compounds and you can arrange them based on whether they're similar to one another. And then you can do a big data experiment where you ask the probability that you would find one type of compound coming up over and over again. And I show an example here of, a scaffold family that was repeatedly discovered in our liver assay. And this is actually very similar to the compound MMV 390048 that Kelly Chibale works on. And so through analyzing these large collections of data, we can get better ideas of what might be a good starting point for anti-malarial drug discovery. The other place that we're using big data is through in vitro evolution and whole genome sequencing. So this is a project that's partially funded by the Bill and Melinda Gates Foundation, and MMV is an important contributor to this. So we are interested in understanding how parasites become resistant, as well as finding new targets that can be used in structure-guided drug discovery. And we take compounds that have come from the phenotypic screens that we've run or for other people have run, for example, have been run at GSK, and we try to identify how these compounds are acting in the parasite. We do this by in vitro evolution. We take parasites, clone them, and then we create, typically over a couple months of selection, we can create parasites that are resistant to the compound, or we can find out that you can't actually create resistant parasites, which gives us a whole new set of compounds that we're interested in. We then sequence the genomes of these parasites, and this can often lead us to the target of a small molecule. We then test the hypothesis, and then we can sometimes validate our alleles. So we have gone through about 1,000, we've created over 1,000 different drug-resistant parasites over the last 5 or 6 years. And we can start putting all of this data together to learn more things that we wouldn't have been able to learn if we only had a single genome sequence. So as we start looking through this large collection of drug resistance, we can start finding all of the genes that pop up over and over again, and might be that we really have to think about if we're trying to create compounds that resist resistance. So the candidates shown on the right side here are genes that have popped up in our selections and are likely to contribute to multi-drug resistance. So we want to be keeping an eye out for these as we move forward with drug development. And on the left side, we have targets that we've discovered through our assays. These are targets that might be taken into structure-guided drug discovery. And when you start seeing very specific mutations in particular transporters, it tells you that this is likely to be something that you want to look carefully at. So that's all I have.
Oh. Well, thanks for that interesting presentation, especially on the area of resistance. So if one can readily generate drug resistance in the laboratory, you might argue that that compound would show limited efficacy. So apart from creating combinations, what type of strategies are you using to create medicines that, as you said, resist resistance?
Yeah, I think this is some place where we need to move into the future, and it's something we haven't been thinking about too much in the past. When I worked at Novartis, there was a big focus on lead optimization and pharmacokinetics and improving the safety of compounds. And considering resistance was generally not considered was not discussed until we moved very, into the end stages of development. And I really think that if we're going to be making better therapies for malaria, but also for things like cancer, we need to be thinking about how quickly the organism can evolve to get resistance. And maybe this is something that should be going into the decision-making during lead optimization instead of at the very end of things. I mean, it's possible that one compound gives you resistance very quickly, but if you made a small modification to it, you would resist resistance because you would have less opportunity, you might have different type of steric hindrance, for example.
Yeah, well, I can certainly see how that would be applicable in multiple different areas, and doing it up front
Right.
could avoid the expense of the rest of the product development pathway and figuring it out later down the road. So that's really interesting. So what do you think we're missing in our phenotypic screens?
Well, as Jeremy mentioned, I think we could really be looking more broadly for compounds that are active against hypnozoites. But I think we need to continue to develop phenotypic screens, because one type of screen may give you a particular type of molecule, and some modification to that screening method can now possibly open up and start giving you a whole new class of compounds. So in addition to additional chemical diversity, which I think we need, small modifications in the different paradigms for phenotypic screening can lead to possibly new classes of molecules as well.
Great. One final quick question. You did talk about data sharing, so can you just elaborate on the value that you see from data sharing? And you showed some interesting examples.
Oh, yeah. I think the data sharing that's happened in the malaria community is really unprecedented. I mean, when I talk to colleagues who work in cancer and on staph and strep, and they're just amazed that we have made so much progress so quickly. And I think the progress that we've made is because we've been able to share our data and our methods and our compounds. And this wouldn't have happened without MMV. They've been very clear about our need to share the data, and I think that's really advanced the field of anti-malarial drug development.
Yeah, that's great to hear. So our next speaker is Bill Charman, who's the Sir John Monash Distinguished Professor at the Monash Institute of Pharmaceutical Sciences in Melbourne, Australia, where he was previously the Dean for 13 years. His research has been characterized by a multidisciplinary and collaborative approach to address major issues in drug discovery, drug delivery, and pharmaceutical sciences. He's published almost 400 scientific papers and communications, and he's been recognized by the Thomson Reuters Clarivate Analytics as a highly cited researcher in the field of pharmacology and toxicology. He's also been quite involved with MMV over his career. He was previously involved in numerous drug discovery projects, many of those that have advanced to clinical development. He was a member of the team that received the MMV Drug Discovery Project of the Year Award on 4 different occasions. He's also been a member of the Expert Scientific Advisory Committee to MMV for over 5 years and is a member of numerous scientific and corporate boards. Bill?
Good, thank you, Dan. And good morning, everybody. I've been asked to make some comment about the importance of collaboration, partnering, and especially about working across different disciplines. It's been very clear to those in the audience that MMV as an organisation is really unequalled in terms of what it's achieved in pioneering a best-in-class means of undertaking this collaborative approach to discovery and development. But if you then read the words that I've got listed in the bottom part of that box there, I wanted to highlight my experience and observations in the way that MMV has done this work, is that their approach, their technique, their means of doing this, I believe, is scientifically generous. It is a best-in-class means of discovering and then advancing drugs that are field-shifting, and we've heard about those from the various countries that are using them. And importantly, this process and funding, selection, it's a tough business, but the way in which MMV has done it, in my experience, has always been one that's been highly inclusive. The means by which projects are selected and then advanced, there is a rigorous project selection process. We heard earlier about the need for transparent, consistent data. The framework that's in place is data-driven. There is no room for emotion when it comes to selecting or funding projects. And the way in which MMV has been able to do this in that transparent and consistent way is also a hallmark of their success. And we've heard from many people today about that network of world-leading clinicians, clinician scientists and basic scientists that have been attracted to the cause. That network, and as Elizabeth commented when she talks to colleagues in other fields, this network is unprecedented. Not only is it an important thing to do, but I think it's the way in which people are attracted to it, the opportunity to contribute. And I think importantly, the fact that there is no monetary basis here for being able to undertake this work. The lack of a profit motive really does allow people to share in ways that they possibly don't do in other fields. It's not only about advancing programs, it's also the financial basis that sits behind it. And the comment that I have at the top of this box here is that the collaborative multidisciplinary model that MMV has designed and has continually improved, I view that model and the framework and the process by which it undertakes its work as one of the essential and highly valuable intangible assets that are on that balance sheet. We often think of profit and loss statements with regard to funding programs, but the intangible nature of the way in which MMV works, it is highly valuable in terms of the balance sheet as opposed to the profit and loss statement. And part of the value of that intangible network is that those external partners, we often think that there's MMV and a partner, but it's actually the partners of the partners. It's the networks of the partners. Those networks can be infrastructure-based or they can be knowledge-based. And the multiplicative effect that occurs here is, I think, what allows MMV to really address questions so that through either its immediate network, its extended network, really, at the best of times, should be no more than 1 degree of separation, one call, one email, one question away from where those answers would be. The final comment on this slide is at the bottom there with regard to the role that advocacy has been important within both university and medical research institutes. The recognition of team-based, or the importance of team-based research, and importantly, the advocacy that's necessary for junior staff members who are part of these large network teams, the recognition of both the impact and the individual contributions they make to their teams, it's been important to highlight the importance of that for their career progression, because it's not common to actually value team-based research to the extent that it probably should be. So on this final slide, I wanted to make some comments around delivery technologies and formulations themselves. There are so many new delivery technologies that are available today. There have been enormous advances over the last 20 years, 10 years especially, in terms of delivery technologies, and much of this has come through the advancements around materials sciences. The fact that many delivery technologies today, the formulation side of things, can be both biologically as well as time responsive. And the exploration of nanostructures, colloids, particularly designed particles, and what I call the sort of nano-everything approach, provides much opportunity with regard to thinking how, as a platform, delivery technologies may be able to support and indeed enhance what Jeremy calls the duration of pharmacological exposure. It's not just half-life that's important here or plasma coverage, it is the duration of pharmacological exposure. And the opportunity to marry that as the goal, thinking back in terms of not changing the molecule that's been optimised to a certain point, or indeed the combination therapy, but what can be done from a standpoint of new technologies to then really be able to attain that duration of pharmacological exposure necessary to meet the relevant target profiles. Some comments about orally administered medicines. A child is not a small adult when it comes to the administration of a medicine. And indeed, a baby is not a small child when it comes to acceptability, the palatability in terms of the administration of an orally administered medicine. The industry, and indeed many organisations, still tend to do development of medicines for children or paediatrics in a linear, siloed manner, based upon the particular therapeutic area. I believe there has to be a better way forward, working in a cross-disciplinary manner in order to develop technologies that are best in class for the administration of medicines, not extrapolated from an adult, but specifically for young children and specifically for babies. I think the best approach here would be along the lines of a shared endeavour, and the hallmark of MMV, that collaboration, that shared network. It is quite exciting to think what might be possible if there was a shared endeavour across different therapeutic areas, but in terms of the technology, to come forward with approaches, best-in-class approaches for the administration of medicines, especially combination medicines, and especially medicines if there is to be a single dose, because that has then particular requirements with regard to drug loading, the size, the palatability. And then finally, from a formulation standpoint, injectable medicines go through various stages of interest. I welcome the resurgence of interest in injectable medicines. It is not only for drugs, but antibodies and vaccines here become important. But in terms of that broader eradication agenda, the ability to stratify the different interventions that are necessary, the approaches that can be taken, there is clearly a role to revisit the opportunities that injectable medicines can play with regard to their role in that broader eradication agenda. Thank you.
Great. Thanks, Bill, for highlighting so many important topics there around the rigors of discovery and development and addressing the next critical question, but also bringing us into this area of drug delivery technologies, which is really fascinating, especially as we start thinking beyond just treatment of acute symptomatic malaria. So I think this will be a critical thing going forward. And I know you think a lot about specialized populations, but could you elaborate a little bit on why the initial development of child-relevant doses is so uncommon and what we might be able to do about that?
Yes, look, thanks, Dan. A comment initially around drug delivery technologies. It's only appropriate to explore what they can offer once you've got the drugs. And if we think back in terms of when MMV started, the drugs weren't there, so delivery technologies were not appropriate to look at. It is interesting to think today with the pipeline, the medicines that are currently available, it may well be time just to then explore what these technologies could then add. If one then considers in terms of special populations and from the pharmaceutical industry model, the market dictates the therapy areas that one tends to go into, and it's generally for adults first. That's where the market is. And the paradigm, whether we like it or not, is that there's typically the development in the various approaches for an adult medicine in an adult dose form. And then over time, and this could be as long as a decade, there may then be the development of or extrapolation to what a formulation may well then be for, let's say, a child. And often it's a smaller version of the adult formulation. There have been attempts in the past to address this, but often those attempts have been in a particular therapy area. And that's probably not enough of an emphasis or an impetus just on its own. And I think what's interesting here, from an opportunity standpoint, is that if there was to be a shared approach, forget about the therapy area, malaria can be the catalyst for it. As long as malaria has got the opportunity to then access the technologies relevant for either a child-friendly or baby-friendly form of administration, all those other applications in areas other than malaria potentially could then be made available to other partners. So I think that where MMV has been innovative in terms of developing a model with regard to the discovery, the development, the access side of things, it would be interesting to think what could be done around that field of child-relevant dose forms or pediatrics.
Well, great. Well, thank you for that, and also highlighting just kind of an area that we could be going into around formulations. The injectables, I think, also provide a whole nother opportunity from a discovery perspective where we were looking for oral bioavailability. And now if we could develop high potency injectable products that through formulations could last a longer period of time, we could really change the landscape. So thank you for that. Our next speaker is Professor Kelly Chibale from the University of Cape Town. Kelly has a very distinct background. He's also the founding director of the South African Medical Research Council and Drug Discovery Development Research Unit at the University of Cape Town, and the founder and the director of the University of Cape Town Drug Discovery and Development Center, also known as H3D. I've known Kelly for several years, and it was no surprise for me to see that in 2018 he was recognized as one of Fortune magazine's top 50 world greatest leaders, and we're thrilled to have you here to talk to us. So please, Kelly.
Thank you very much, Dan, for a very kind introduction. I'm going to speak about three main topics here. The first one is what you see on the screen, which is what are the drivers of innovation in African science and medicine. And the second point is I'm going to exemplify a project that really illustrates what I believe are some of the drivers of innovation in Africa. And thirdly, I'll finish off with just really highlighting how this new model of doing things in a network of partnerships that MMV has really pioneered, and Bill Charman was referring to just a little while ago, has really impacted the way that we do things and how this is having a tremendous impact in other areas that also people can learn from in other disciplines. So, firstly, what are the drivers for science or innovation in Africa? So from my perspective, it's three things. The first thing, it's a no-brainer that Africa, of course, is a very, very, it's a continent with enormous health challenges. And I think it's also a no-brainer that is vital for African scientists like myself to really get involved in contributing to finding solutions to these problems, which are not African health problems. They are human health problems. And that's the first point. The second point is another fact, which is that all along, for decades and decades, Africa has been reliant on innovations that happen in the global north. Africa has been a beneficiary of these innovations. And of course, if we, as Africans, are going to be partners, we also have to contribute so that other people can also benefit from our contributions. The third driver, in my view, is actually using science for development. And I will exemplify this in terms of the way that we've gone about doing things and how we're changing the landscape and really showing in a small little way that we can use science for development. So those are the three things that really, for me, are the drivers for science and innovation in Africa. Now, what about how do we exemplify this? How do we exemplify that Africans should, of course, not on their own. We cannot do it alone. Just like if you are Liverpool Football Club, you will never walk alone. So really the point really is how do we as Africans partner with international partners to contribute to solving these human challenges? And that's the story, of course, of the project that we've done with MMV that you saw in the morning session that led to the discovery of MMV 390048. Elizabeth mentioned this a little while ago. Of course, this is a remarkable story, remarkable in many, many respects, but also shows you that we in Africa can lead international efforts. This is not something we did on our own. This is not something we could have done on our own. But the point is that we led this international effort with many partners, MMV, Swiss Tropical, Sergio Whitling here, Monash, CDCO with Sue Charman, Vic Avery I mentioned in the morning when we started, Syngin, GSK, many, many people that I can tell you were partners in this story. But the key message that this early discovery of this compound was led out of Africa. The early clinical development of this compound was also led out of Africa. The phase 1 by Karen Barnes at University of Cape Town. And you heard this morning from our colleagues in Ethiopia, Daniel and Rizema doing the phase 2 trials in Ethiopia. Again, exemplifying that we can also lead international efforts. MMV 048 is not the only story. In fact, I'm much more proud of the fact that we've really been able to build critical infrastructure that never existed before. That we've grown the portfolio of projects within malaria and brought in multiple partners as well. We've expanded not just to malaria, but also to TB, and much more recently, antimicrobial resistance. We've shown a portfolio of projects at different stages of the value chain. And of course, this is not a once-off. It's to do something that is potentially sustainable. So apart from building the critical infrastructure, the technologies, and on the road to training a critical mass of African scientists, going back to what David Reddy said in the morning, we can prepare the next generation to really tackle malaria eradication. My final point is this model. When we started in 2009, and I said this morning, the only thing I had was chemistry, 5 postdocs. We've grown to an 80, 90-person team across multiple disciplines in chemistry, biology, and pharmacology. But this model was about saying, what can we do with what we have without wishing we had everything? And how can we tap into our network of partners that MMV put together? So this model, as you heard this morning, so this is a model of industry, academia, and PDP partnership with a very strong mentorship component. So I mentioned Mike Whitty this morning. So that mentoring expertise from Mike as an experienced senior scientist, not just mentoring me, but my team, is a very powerful cocktail of how you can build capacity while getting the job done. Thank you very much.
Great. So thank you, Kelly, for that inspirational message, and certainly having Africans lead on African problems is going to be vital going forward. And your leadership in this area has been fantastic. Maybe you could share with the audience some of the lessons learned that might enable other African partners to replicate what you've done in Cape Town, because that's quite an operation you have there.
Sure. I guess, they say it all looks easy when you know how, but of course it's never really easy when you start. So I really would, when I look back and with the benefit of hindsight and experience, I really would say the following. So I think the first thing, which is really something that bedevils my continent, is infrastructure. When I say infrastructure, I'm not just talking about a physical building. Infrastructure also means systems. Can you even order chemicals that arrive on time? Because drug discovery, you make decisions about which compound to make or to step a project. So I think infrastructure is number one. The second lesson I think I've learned from this project is the importance of having a globally aligned project. And this is where I think the role of MMV was really important. That apart from avoiding duplication, that you do something that is really, really relevant globally, that you can then tackle this challenge collectively, as an international partnership. The third lesson I've learned, again, I go back to the point I made earlier. So it's not about having everything that you need at the start. It's focusing on what you can do well in your environment. And then you tap into a network of centers of excellence or network of partnerships, which Bill was talking about earlier. And I gave the example repeatedly of Sergio Whitley at Swiss TPH, so Charman at CDCO, Sinjin, of course, Vic, every at the beginning. And of course, you get the job done that way. The fourth experience, of course, is having experience on the team. So it's not just Mike Whitty as an experienced mentor on my team, but also MMV employs people who are themselves ex-industry people. Jeremy Burrows here was the first director of my project, then David Watterson, and now James Duffy. But all these people bring industry experience that really becomes important. And finally, money. Money is the last thing you mention, because money will follow good infrastructure, money will follow a good project, money will follow a good network, and money will follow experience.
Well, thanks for those points. And I remember my first visit to Ethiopia, and I went to what I call the laboratory graveyard. And it was in one of the largest medical schools in Addis Ababa. And they said, here's a bunch of well-meaning donors who provided us with all of these machines, and we have no way to get the reagents. And it was just a massive basement full of, essentially dead equipment. So that's a valuable point. And I think it speaks to how do we have that communication, and how do we open up the lines of communication so that we're not having these types of graveyards? One other quick question is how do you think we can optimize the scene for discovery and delivery of drugs to the African population?
So I guess because historically, whether it's drug discovery or clinical development, et cetera, so historically, this has occurred in the global north. So the discovery, of course, if you're in discovery, one of the things that, of course, we do is if you understanding or you front load drug metabolism studies, you access whether it's microsomes or hepatocytes that you buy commercially, and I'll come back to this point a little bit later. And of course, when we buy these microsomes and hepatocytes commercially, these, of course, are derived from Caucasian or Asian populations. So at the level of discovery or preclinical, we don't really have the tools that will allow us to, when we are optimizing small molecules in the value chain, that we can really prioritize those molecules that perhaps have a better predicted pharmacological profile in African patients. In other words, rather than using these Caucasian or Asian-derived systems or tools, that I think is important now to think about doing this for the African patient population, because that kind of data really impacts on whether we do human dose predictions or stratifying patients for clinical trials. Right. So that's one extreme. The other extreme is really the low volume of clinical trials. Because of such a low volume of clinical trials, and of course statistics say less than 2% of clinical trials that happen globally happen in Africa. So therefore, you think about it, the clinical trials in volunteers or patients are done in the global north. So the dosage intervals, et cetera, are optimized in those places. And then the therapies are brought to Africa. And of course, what we don't factor in are extrinsic factors, physiology, genetics of the African patient population, or even those actually intrinsic factors, those I mentioned, genetics and physiology. Extrinsic factors, the practice of medicine, traditional medicines. In Africa, when does the patient present themselves for treatment? Is it at the beginning or is it late? So, bottom line, I think it's really important that we increase the number of clinical trials in Africa. There are many ways we can talk about. Africa has to be an attractive destination for the global pharmaceutical industry, who are the major funders of clinical trials. But finally, I think African governments must demand local clinical trial data, as it happens in other countries, because then that will force us to do more clinical trials.
Yeah. Well, great. Well, thank you so much for those insights. And our last panelist here is Dr. Lutz Hegman from Novartis, and I think it's great that we have an industry sponsor here because, we talk about sustainable development goals and all of these types of things, and in reality, many of those will never come to fruition if there's not a true partnership. So just like there's a diversity of folks on this panel, I think it's great to have Lutz here representing industry. He is the Chief Operating Officer of Global Health at Novartis and oversees global health operations from drug discovery through development to implementation. This includes Novartis's flagship programs in malaria, but also in leprosy, sickle cell disease, and Chagas disease, as well as the continuous development of the established medicines portfolio, specifically with emphasis on underserved populations. He holds a medical doctorate degree and a PhD in molecular pharmacology. He's trained as a dermatologist, somehow made his way to malaria. He's also a fellow of the Royal Society of Tropical Medicine and Hygiene and serves on the executive board of the Swiss Alliance Against Neglected Tropical Diseases and is on the board of trustees of the Novartis Foundation. Lutz?
Thank you very much, Dan. Ladies and gentlemen, let me start by adding my congratulations on behalf of all of us at Novartis to those many congratulations you have rightfully so received already on this milestone anniversary. And I think it is amazing to see 20 years of unwavering commitment, 20 years of a very clear focus to advance science and medicine to address unmet medical need, and 20 years of forging new partnerships across a very diverse spectrum, all in the interest of replenishing what was at the time empty malarial pipeline. So congratulations, MMV. At Novartis, I would say that the fight against malaria is part of our DNA and has been for many years. And I'm not going back to 1939 when we first discovered DDT that was mentioned and its benefit in South Africa and other parts earlier today. But over the last recent 20 years, we've been able to provide 900 million of Coartem treatments to patients in disease-endemic countries. And we are very proud and privileged to have been working with MMV for the last 15 years. And as you heard in this morning's session, one of the great accomplishments was the development of Coartem dispersible, a child-friendly formulation of Coartem together with MMV that was introduced in 2009. So our interest in malaria intersects with a strong commitment that we have on child health. And in fact, I was in Ghana with our CEO last week to launch the first holistic disease management model for sickle cell disease, which is another major killer of children in those countries and obviously has evolutionary crossroads with malaria. But beyond that, and we spoke earlier about the importance, 50% of all of our products carry a pediatric label, and we are running about twice as many clinical trials in children as any other sponsor does globally. So there is this commitment, certainly both on malaria, but also on pediatric health. But let me go back to malaria. Last year, we announced a commitment to invest $100 million over the next 5 years to advance our malaria pipeline, both on a discovery side. We have the dedicated Novartis Institute for Tropical Diseases under Thierry Diagana in Emeryville that many of you collaborate with, but also on the clinical side, where, again, we are very privileged to partner with MMV in the advancement of 2 lead candidates, cipargamine and ganaplacide, that collectively with other combination products can address a variety of unmet medical needs in malaria, from severe malaria, uncomplicated malaria, and potentially even chemoprophylaxis. And here it is so critically important that we leverage partnerships, and we are particularly grateful for the partnership with MMV, which not only helps us with strategic and operational guidance, but certainly also helps, and I think it's fair to mention that, financially in bringing those assets forward, which would be a difficult commitment despite a strong desire to do so from any pharmaceutical player. So partnerships, to me, are at the heart of what we do, not only in global health, not only in malaria, but in global health at large. And it's critically important that all players, be it government, be it the public sector, in industry, in universities, institutes, very strong collaboration with the Swiss Tropical Institute, but also the private sector work together to achieve a common goal and a shared objective. So more recently, we have been fortunate to be working now together with the European Developing Country Clinical Trial Partnership, which also addresses a critical component that Kelly had mentioned, to build clinical trial infrastructure in Africa and to bring more clinical trials to the African continent. There will be a focus on malaria together with MMV in a recent grant that was awarded, but also with other private sector players, as we discussed at the Grand Challenges meeting just a week ago in Addis Ababa. So I think that partnership notion is critical, and we are ever grateful for the role that MMV plays in this ecosystem. Let me close by making one final comment on the other theme that was prevalently covered here in this panel, and that about data. I think we need to even step up further when it comes to sharing data, when it comes to considering data not just as a means, but as an asset in its own right, and extending this beyond drug development, where the benefits are very obvious, and Elizabeth had talked about those, but also considering how can data inform programmatic implementation, how we can be more precise in using the right approaches as we are targeting interventions, and that would be my wish for the day, adding to the celebration. Thank you very much.
Great. Well, thank you for that, and I certainly appreciate your comments on data sharing. Just one brief question. Could you talk about product development partnerships and why they're important? I know at one point, when I joined the Global Health Committee, I'm like, well, what are these PDPs and how do they work? And I was an industry person, so it'd be great to get your insights on that.
Well, I mean, I would say the short answer is if PDPs didn't exist, it would be timely to form them. Fortunately, they do exist, and I think the benefit is manifold. I touched upon a few of those. To me, it really opens the perspective of each and every player and help connect the right players. Specifically on strategic, scientific, and operational excellence, you have the ability as PDPs to look across all different industry players. You can connect industry players with public sector players, whereas we, representing a specific sector, of course, can only look into our own immediate ecosystem, and I think we are missing a big benefit here. And then beyond that, the advocacy piece, but also the co-financing, which I think turns every donor money into probably 3 to 4 times what is being invested when it comes to output. In an area of global health where there is a scarcity of resources, no matter where we look, I think we have to spend every investment very wisely, and PDPs help us do that.
Great. Thank you very much. Andrew?
Great. So, well, thank you all very much. Thanks, Dan, for leading it, and a great panel to finish the morning. I must say, I've always thought product development partnerships shouldn't just be there to sort of fill in the gaps that for-profit pharma has sort of left behind for market failure reasons, but actually have the potential to be a field ground for experimentation and innovation in precisely bringing together in many ways and forcing collaborations that the for-profits alone don't do. So it's great to hear that in practice. But do please thank our panel. Great. I think you probably want to, you can just stay fine.